Hello everyone. Today I'm going to talk to you about the vast community of microorganisms that live in association with us. Every surface, every cavity of the human body is colonized by this complex community of microbes. From the day that we're born, we live in close association with them. As we are now just beginning to apply the tools that came out of the genomics revolution to understand them, we are just starting to understand that they are critically important for our health, and they also play important roles in disease. The estimates are that each of us carry hundreds of trillions of microorganisms around with us every day. There are 10 times the number of microbial cells as there are human cells associated with any individual. So only 10% of our cells are human cells. If we collected them, they would weigh about 1 to 2 kilograms. When we think about it, we are just sacks of microbes walking around. We are more microbes than human. In 2008, out of the NIH came this bold project idea, what is known as the Human Microbiome Project. It was really a follow on the Human Genome Project, and this idea was to create an interdisciplinary group of scientists to study the human microbiome. So they went around and recruited 250 volunteers, equal in number of men and women, and have them agree to be swabbed and scraped and probed over a 2-year time period to collect material to better understand these microbial communities that live in and on us. We've learned that each individual has a unique ecosystem of microorganisms made up of thousands of different species of bacteria and other organisms such as fungi, yeast, single-cell parasite, and viruses. Moreover, it seems that each location of the body has its unique combination of microbes. And it really seems that we've co-evolved with these microbial species so that the microbes that are present in any given place in our body carry out specific and important tasks. So each of us is unique in terms of our microbial ecosystem. They are another molecular fingerprint of us individuals, if you will. There was an interesting study published in 2012 that collected microbes from the fingertips of volunteers and also collected microbes from computer keyboards. And without exception, they could accurately map the owner of the keyboard back to the keyboard. That's how unique they are to us. What's also important to know is that these microbes have many different and specific functions in our body. We first acquire them at birth by passing through our mother's birth canal. In the early stages of life, they play an important role in educating our immune system and teaching them to identify them as self and not as foreign invaders. And this education process can only be done in infancy. And in fact, they are so important that doctors in modern hospitals today will now take a vaginal swab from mothers to inoculate their C-section born babies to make sure that they grow healthy. Most importantly though, they help protect us from infections from pathogens, especially in the early stages of life. We already know that babies born by C-section get a very different inoculum of microbes than those babies who are delivered through a vaginal delivery. And these differences tend to persist for months. We also know that infants that are fed formula instead of breast milk end up with very different microbes. In fact, breast milk contains microbes that need to colonize the GI tract of babies. And the breast milk also contains fruit oligosaccharides that aren't there to feed the babies, but are there to feed the microbes that live in the infant's GI tracts. In the GI tract, these microbes help us harvest energy from all the foods that we eat, breaking down plant polysaccharides that we ourselves can't digest and provide us with up to an extra 10 to 15% more calories. They produce vitamins and metabolites that are essential to maintaining healthy bodily functions. They provide substrates for synthesizing hormones like melatonin and epinephrine, or for synthesizing important biochemicals of the nervous system, such as dopamine and serotonin. They also ferment some of the food we eat, producing every day the equivalent amount in alcohol as in 1 can of beer. And as we all know, some of these bacteria also produce gas. And some people have strains that even make these gases flammable. Please don't try to find out what strain you have at home. It can be quite dangerous. So, what are the factors that influence this microbiome? So, to name a few: stress, sleep quality. Moreover, studies have shown that with increasing urbanization, decreased contact with nature and with animals seems to matter. There was a study showing the differences in microbiome composition between matched for age and gender US urban children and children from Malawi or New Zealand tribes. What's immediately apparent is the urban US children had far less diversity in species in their microbiome. The fundamental principle of an ecosystem is that it has to be highly diverse in order to be stable, resilient, and able to return to a balanced state after a perturbance. There's something called the hygiene hypothesis, that we're living too clean may actually be detrimental to our health. Kids need to expose themselves to as many microbes as possible in early life to increase the diversity of their microbiomes. And kids are instinctively experts at this. Kids put everything in their mouths. They are expert self-inoculators. But these days, as soon as Johnny's pacifier falls to the ground, it is disinfected by mummy with Purell before it is given back to him. Antiseptic sprays and lotions are in every purse. Studies have shown that getting a dog can significantly increase microbial diversity in children and reduce the rates of asthma. So go get a dog, it's healthy. Now, perhaps most interestingly, the increased prescription of antibiotics in early life can lead to an altered microbial composition in children. And this may predispose to certain illnesses later on in life that are related to changes in microbiota. In the past 20 to 30 years, we've seen a very sharp increase in the incidence of allergies and food allergies and autoimmune conditions like asthma. Actually, I think for asthma, there's been a 4-fold increase. So what does this have to do with the microbiome? Well, studies have shown that if you expose young mice to antibiotics, you can actually mimic the allergic phenotype that we see in humans. And when you look at the diversity of microbes in these mice, they have lower diversity than what you would see in mice that weren't exposed to antibiotics. And studies have shown that these differences seem to persist into adult life. Also in adult life, antibiotic use is a huge factor. Not only do they kill the pathogens and foster the development of resistance of those pathogens, but they also destroy many of our commensal microbial colonies that are actually good for us. And it's good to remember that these antibiotics don't only come through your doctor's prescription, but also from the meat and dairy products, because 80% of all manufactured antibiotics in the world are used in animal agriculture. So of course, one of the main factors is diet, and today's supermarkets are flooded with delicious but processed and rather unhealthy food. And what we eat every day, or more importantly, what we don't eat, has the greatest impact on the microbiome. All the different species of bacteria that live in the GI tract all perform different functions. And to keep them all happy and thriving, we need to feed all of them. That means eating a very diversified diet. If you only eat a diet of potato chips, you will encourage the growth of the bacteria that are good at breaking down potato chips. But all the other bacteria that produce all the vitamins and other molecules that are really good for you will start to die off, and perhaps in time you'll start to develop some kind of disease. For me, what's been the most interesting part of this growing body of microbiome research is that studies are finding more and more associations between the microbiome, its diversity, even the presence in the gut of some specific strains of bacteria, and how this ties back to the broad spectrum of diseases. Associations have been shown between the microbiome and autoimmune diseases such as Hashimoto thyroiditis, multiple sclerosis, rheumatoid arthritis, inflammatory bowel disease, also with metabolic diseases such as obesity and diabetes, psychiatric illnesses such as anxiety and depression, and neurodegenerative diseases such as Alzheimer's and Parkinson's. The evidence shows clear associations between changes in our microbiota and these different disease states. For now, these studies only show associations, but there are now studies underway to address causality, because in medicine, research isn't actionable until we have the holy grail of causality. So this brings up the interesting question of therapeutic targets. How can we use this information to improve treatment of disease? Well, we've had probiotics around for a while, but for a long time, these have been regarded as mere nutritional supplements, and the manufacturers are not held to any scientific efficacy standards, and they don't need to undergo anything like FDA approval. But this new information has spurred a new interest in probiotics, and we are starting to carry out rigorous scientific trials on probiotics and how they can be utilized as therapeutic options in various diseases. So a few years now, modern hospitals have started to integrate a procedure called fecal transplants into their array of therapeutic options. And I'm going to get very graphic now. Basically, it involves taking stool samples from healthy donors, putting these samples in a blender and making a poop smoothie, and then administering this solution to a sick recipient via a nasogastric probe or an enema. Now, this procedure has a very serious ick factor to it, but it's actually been shown to be the best treatment and can have up to a 95% cure rate for treating patients with antibiotic-induced diarrhea or Clostridium difficile diarrhea, which can be lethal, especially in frail and elderly people. In the lab, fecal transplants in mice have also shown to have an effect on the development of obesity and diabetes. A recent study showed when a fecal transplant from a fat diabetic mouse to a skinny non-diabetic mouse was done, the skinny mouse would get fat and diabetic, and vice versa. The transplant of the microbiome from the skinny mouse to the fat mouse would rid the fat mouse of its extra pounds and diabetes. So in humans, there was even, I think, a case in the US that was reported where a sick mother received one of these fecal transplants from her healthy but a bit overweight daughter, and she became healthy, but she also subsequently gained quite some weight, and so much so that she ended up suing the hospital for this. So the success of this procedure has sparked interest in creating perhaps a more palatable method of administration, and there are now some labs that have come up with capsule forms, or as they like to call them, crapsules, with the new formulations of probiotics in order to repopulate the gut microbiome, as they like to say. So I can talk about the microbiome and all the wonderful things it does for a very long time. But if I would have to sum it all up, it would go along the lines of something like this. The microbes are your friends for life, and take care of them and they will take care of you. And don't forget, diversity in what you eat will lead to diversity in your gut. Thank you very much for listening.